Yeah, thanks a lot. And thanks to LGC for the invite for me to come here today. I think Eugene has really done a pretty good job in stealing my thunder and talking to you all about the technology and the things that we do. I talk to Eugene quite a lot across on Zoom in our process development, and especially with our close relationship and development with the R&D team in California. So I'll share some of the results in our slides, but mainly we'll talk about a little bit about the company BioAcumen, what are the things that we do, and the recent developments, especially that we have during the pandemic. So as a company, we are really very young. So I just share a bit on my background. I was actually part of A-Star before, and that's really long ago, about 13 years ago. I was actually working in a lab that was very focused on translational research, producing PCR kits, right, and working very closely back then with NUH and TTSH to convert a lot of the multiplex PCR assays that we were doing into actual clinical usage. And if you think about 13 years back, right, we are actually already producing 5-color probes, 5-color multiplex tests in a single assay. So at that time, it was really exciting times for me. And back then, when I see those technology from the lab that I was working for being commercialized by local companies, right, it was really exciting. But after a while, I was like thinking, hey, maybe I could actually be having my own company, right? And actually selling those assays myself. So that idea actually stuck with me. And finally, in 2017, I actually did the bold move, right? To actually have my own company called BioAcumen Global. And at that time, I can share a little bit about when we first started out. We took a very small cubicle office, 2 meter by 2 meter. And in it, we cram every single instrument that we can get, from the centrifuge to a ISO class 4, very small fume hood that's for only one person, to a combi freezer where I can store my 4-degree and -20 temperature sensitive reagents, all into a 2 by 2 square meter office. And that was actually enough for me to start taking orders and doing all kind of different real-time PCR assays. And the weirdest request I remember at that time, because nobody will ask us to do IBD kits. So we had to take a lot of other orders that nobody wanted to take. So one example, very weird order from Hanoi. This professor from a natural study ecology university was actually asking us to customize a test kit for crocodile lizard. I was like, what on earth is crocodile lizard? Never heard of that before. So I did more research and I actually found out that crocodile lizard in Vietnam is actually a national treasure. And they were quite concerned of the animal actually going extinct. So they were going around in the forest, picking up different types of feces, fecal matter, and actually doing eDNA testing to try and track and trace where are the likely places that they might be going. So we did our research and we're actually quite glad to find that there are actually public database, right, that contain DNA for crocodile lizard, and we're able to make a cybergreen-based assay and lyophilize that for the customer. But that actually pushed me into another problem, because we can manufacture, but the customer was asking us for lyophilized assays, and we had to outsource that. And so my next question was, is there anyone in Singapore that's actually doing this kind of contract manufacturing for local startups? And there wasn't any, so we actually had to depend on another company overseas and to send the primers and probes over, get it lyophilized, send it back to us, and I had to carry in my luggage to my customer in Hanoi. So that was actually a lot of cost involved, and in the end, we earned a bit, but not that much. So it pushed us really to think about, hey, why don't we just have the whole suite of manufacturing capabilities, right, in-house, right here in Singapore? And the pandemic was actually a big push for us. It was like, okay, it really makes sense for us to do this now. So we actually moved into our new facilities relatively recently, July 2020, into a new office, and we make sure that we get a big enough space this time to actually go for ISO 13485 certification. And very quickly in September the same year, we got that. And we communicate a lot with HSA because we at that time we already had the design ready for a COVID test kit for 2 gene detection for N gene and ORF1AB. And HSA was actually quite helpful in with us to actually took our request quite urgently and issue us with the provisional authorization. So with that, moving quickly into December in the same year, we put in additional R&D resource and budget to add and expand for freeze drying capabilities, which has also in 2021 been added under to cover our ISO 13485 scope. So very rapid developments. You see in a short time, then we actually opened a new office in Thailand, which has already officially started last year. And this year in April, right, we're actually one of the first companies to achieve a full-fledged authorization, converting from a emergency use only to a full-fledged HSA-approved SARS-CoV kit. So as a company, what do we do? And very typical for startups, especially biotech startups in Singapore, right? Because we are quite hungry. So we try to do bits and pieces of everything. Any business that you can find. Normally, people that know me, I won't reject. So I will usually accept. So we do distribution, we do manufacturing, we do custom PCI designs, all under a ISO 13485 quality management system. But as the company matures and move forward, right, I really do see us really concentrating a lot on contract manufacturing services and custom PCR design for our clients. So our expertise really centralized around molecular diagnostics, especially using PCR technique. And we have proven applications in many different areas, especially in veterinary diagnostics. We're actually pretty strong in Singapore. In fact, I can confidently say that we're actually the biggest provider for veterinary diagnostic, but especially in terms of pathogen testings, right, for vet clinics, especially for companion animals in Singapore. And we do also for human infectious disease and environmental testing. So in-house manufactured kits, as I've shared, because of the pandemic, we have actually went into our own brand for COVID test kit. And during the Omicron new variant last year, somewhere around end of November, December, we actually took the published database on GSAID very quickly, right? And within 2 weeks, come out with a new Omicron detection kit. And back then, it was only the first variant, which is the BA1. And in February, when there's BA2, we actually managed to find what are the mutations that can differentiate between the two. We actually helped Oxford Clinical Research Unit in Hanoi to actually come up with a customized kit for them to do surveillance studies to differentiate between the BA1 and BA2 subtype detection in Vietnam. And moving forward, we see respiratory virus detection as something that is going to be important, especially during the winter season. So we actually put in some R&D, right, to come up with a winter virus assay that detects not just for COVID, but also for influenza AB, RSV, AB, and perhaps future viruses that may be a concern, right? We'll build this in the panel in one single assay. So, as I was mentioning at the start of the company, right, we were facing a lot of difficulties because part of the scope of work, sometimes we need to outsource to other countries or to other contract manufacturers. But beginning of 2020, we felt that we should have everything in-house. So from the design and development of a NDX kit to the production, to the storage and distribution, we have actually worked very, very hard to make sure that within our facilities, right, we will be able to do something from end to end, all the way from when the sample comes in, from raw materials until the final packaging is done. Our scope will be able to cover for that. And our current PCR kit manufacturing capabilities, right, as I mentioned, from the very beginning, from in silico design to product verification, validations, as Eugene has shared, for example, LOD, sensitivity, specificity determination. And then finally, when we have cast that in stone, we will pass that to our production for the SOP that's needed for the production protocol. And lyophilization is actually something new since 2021 that we have added into our ISO 13485 guidelines. And finally, the packaging, and of course, the clinical evaluations that we have with our partners all over Southeast Asia before we arrive at the final products. So it's pretty comprehensive ISO 13485 certification that we work very, very hard to achieve. And we are keeping this open. So let's say there's anybody who's thinking of wanting to do a startup, or a startup that already has a product that's being designed, but they are missing ISO 13485 coverage for production or packaging. Anyone can actually approach us, right? We can sit down and we can actually plan out. And you do not need to take up the whole service. We can actually go modular. Let's say you only need lyophilization and packaging. We can certainly customize that kind of service for you. So as Eugene has shared, there are certain technical considerations for lyophilizing of IBD products. So for us, I think what is really critical is at the start, we need to establish what is the collapse temperature. And then we believe that this is actually one of the most important steps for the whole process. So in the picture here, what is shown is actually one of our first few batches. So we create these little beads called lyo beads using liquid nitrogen. And it took us actually quite a while to establish what is the critical temperature that can actually withstand the primary drying without collapsing. So there are certain technologies that you can actually use, pretty expensive instrumentation from differential scanning, calorimetry, freeze drying microscope, and dielectric resistant analysis. Some common methods they can use to establish critical temperature. Or if you want to do it by putting in your primary drying stepwise method, so slowly increasing the temperature or the pressure, that can also be done to establish your collapse temperature. But it's an important step that you need to go through first before you can actually go into the actual production. And as Eugene has actually shared in the previous slides, a lot of important components within a PCR reagent that you want to freeze dry that you need to consider. And buffer is especially important. We would definitely recommend Trees buffer. So as we need to undergo it's very sensitive to pH fluctuation. So anything that has phosphate buffers are definitely not recommended. Bulking agent, another very important component that's actually needed to give your final product structural support. So very common reagents such as sucrose and trialose actually being used, but there are plenty of other disaccharides, right, that we have tried, and it's also possible. Surfactants are also another important component to help against aggregation and reduce adsorption. For example, Twin 20 detergent. So why go through all this trouble? So many things to optimize and consider. So I can share some of the stories so if you notice our timeline, it's only in December 2020 that we decided to set up the lyophilization facility. So what happened before that? So at the start of the pandemic, when we started shipping our first version of assay, they were non-lyophilized. there were a lot of shipping disruptions. We had huge shipments in cartons, stored in dry ice, stuck at various, say, country I or country T or country V in ASEAN region. And the custom officers were sitting there waiting for, say, some missing documents. And different countries have different things or regulation that you ask for. And the dry ice that we put on probably can last us for about 3 to 5 days. And sometimes because of the custom office or because during weekends they go on off, you could have goods that are being stuck there and the dry ice completely are gone by the time it passes the custom. So it has happened to us before. And disruptions like this during the pandemic were very, very commonplace for us. And to add on the cost of shipping because of dry ice, not just because of the cost of dry ice, but because of the weight of dry ice, and because it's a dangerous good, there are certain specialized service that you need to get to actually ship your things over. So all these are really, really added to very, very big headaches for us when it comes to logistic planning. So we really, really believe that pre-formulated lyophilized reagents, right, minimize a lot of headaches that they are going to have. And in terms of the end user, right, when we have these products and when we ship the first prototypes over, we realize that for end users, it's very easy for them to use because everything comes pre-mixed. So they just need to add 20 microliters of their purified sample, and it helps to reduce a lot of potential contaminations that may happen during a master mix step. So the end users love it. It's easy for us to ship. So really, lyophilization was the no-brainer for us, for the company, and it's something definitely that we have to get. And we did quite a lot of accelerated studies also to test and make sure that our end products are able to reach at least 1 to 2 years shelf life at room temperature storage. So from something that you see in the middle, the cake format was something that we did when we first had the lyophilization facilities. Then we slowly move into other multiple different formats because we're also doing quite a lot of contract manufacturing for customers that are coming to us. And each customer may have their own requests or asks. And we are seeing more and more customers that are actually requesting for lyo beads. Not to reveal too much detail, but as I mentioned just now during the panel discussion, there are a lot of companies that are trying to develop their own POCT devices. And when it comes to POCT devices, they have their own device and they have their own plastic consumables. So sometimes that will mean that the lyophilized cake cannot be fixed within a 96-well plate. And we have to put it in a format that is easy for them to actually transfer from a container into their cartridges. So we do see a trend that lyo beads as something that has been one of the top requests from our customers. So another key consideration for us, right, is at the end of the day, we can have the workflow, we can have the facility, but we can't possibly, as a startup, take care of every single components. We do need commercial suppliers or reliable partners that can work with us. So master mix that's needed and the reagents that's needed is really important for us too. And we do need to work with someone who can actually consistently produce things that are reliable and consistent lyophilized products. So some of the considerations, no issue with lot-to-lot variation. So when we sign on a new commercial partners, right, these are the things that we do. We produce different lots with different technicians and we compare them in terms of the standard deviation, stuff like that. We test for 1 year shelf life at room temperature, both in accelerated and real-time studies. Other key considerations, for example, good application support. For us, no R&D process is 100% smooth sailing. So whenever there's obstacle along the way, right, expertise, for example, I can just call Eugene, and he is able to get on a call and assist us on this, right? That would be great. And of course, pricing. As a startup, we are always very price sensitive. So if there's things like tier pricing that we can work on, that will be good for us and our customers downstream. So we were approached by LGC Biosearch Technologies in late 2021, because at that time they told us that they wanted to develop their Lyo-ready reagents, and we're lucky to actually work with them on this. So even things down to the Mg2 plus, the different sugar bulking excipients, right? We actually, it was not just a service for them, but we also gain quite a lot of experience and insight into the products and on how to improve something from scratch. So you can see actually some markings, right, on the plastic bags for these beads. So these are, we are actually working at that time, at the start in November, working on what are the correct Mg2 plus concentration for the solution. And when we finally set on a composition, we decided on a final concentration of 10% sucrose to be tested with our in-house SARS-CoV kit. And we wanted to do a robust testing of this prototype reagent. So what we did was we separated the reagents into 4 different batches, and we produced more than 600 beads for each batch. And we bought actually a commercial control varicella from Spain to carry out the test of the reagents. And there was actually an additional test on the stability of the lyophilized product. So to go through quickly, there's 4 batches that was being done. This is the results for the very, very first batch for the composition that the final composition that we decided on. So there's 2 types of curves, both for SARS-CoV-2. So in blue is ORF1ab gene, and in red is N gene detection. So 2 different targets on the genome for SARS-CoV-2, and you can see very, very similar CQs for both, slightly better for the N gene. But when you look at the standard deviation and the percentage CV, right, they're both very good values for something that we have done for the very, very first batch that we produced. So batch number 2, carry out a few days later with a different technician, still very good results. We saw standard deviations all below 1, except for the ORF1AB, which is slightly above 1, but still good results. And then when we move into January, we repeated this with the 3rd batch, good standard deviation and percentage CB again. And finally, when the last batch that we did, you can see even for the RFU values. So by the time we have really refined a lot of our SOP for these reagents together with these test kits. So by the time we were doing the 4th batch, the technician is actually quite well trained already. And you can see in terms of the average CQ, right, very, very high, very, very good sensitivity. And in terms of the RFU, very good fit at the top, near to 700. And finally, we produced another additional batch. This was done on the 26th of January, and we store it at room temperature, leaving it around. And we open the bag and we test it on the 2nd of March. And this was done with our MS2 assay. And this was the original QC results that was collected in January. So 35 days apart. So we took the MS2 stock that we purchased from Roche and did a serial dilution. And you can see even down to -8 dilution when you compare the 2nd March and the original QC results. CT value are very close of 32.74 and 32.24. And the RFU values, right, essentially the same. So very, very good results that we have. And we are still keeping some of these reagents and continuing the study for second month, third month. So every month we'll be doing this. So in conclusion, the rapid fire LyoFlex one step RT-qPCR 5X master mix, not too bad. We concluded it was robust enough for a lot of our different real-time RT-PCR reactions that we have produced in-house. And the 5X master mix to us was actually quite attractive because they actually gave us the flexibility to make our choices for excipients, and there's enough volume for us to play around with different things that we want to mix. Good results, as I've shown, with little lot-to-lot variations, and it was quite incredible because it was very first batches without any much for optimization needed. I would say, having tried a lot of the other products, is quite well priced compared to alternative options on the market that might be older or market leaders. Feel free to check with the scientific resources and LGC team here. And if you like to do contract manufacturing with us, you actually do get a 30% off all lyophilization service at BioAcumen Global if you were to use their products. So just a bit of selling over here. And lastly, in my presentation, I just want to conclude a bit on the point of care diagnostic devices. Because as I mentioned, because of the number of contract service that we have seen and people are asking for lyo bits, although downstream, they didn't reveal to us what they are doing, but we believe that the pandemic has actually accelerated the need for fast and easy to use platform that can carry out near patient testing, for example, at immigration checkpoints. People and hospitals and labs are looking more and more at direct samples to result with minimal hands-on and training. And there's a lot of demand for higher throughput with ease of transportation and storage. So because of all these demands, lyophilization has actually become a key manufacturing process for this assay used on these platforms. So the lyosphere that we produce, right, is actually a pretty specialized service here in Singapore, and it gives customers actually a chance, right, to put all these beads into their own customized cartridges, which is excellent for POC devices. And all these will be assembled, packaged, and distributed, covered under our ISO 13485 guidelines. Alternatively, if you like to carry this out yourself, so if you don't want us to package the cartridge, we can actually do specialized packing in inert gas with dexicants provided and sent to customers, and they can carry out the work on their side. So just some of the proven track record that we have in less than 1 year of launching our lyophilized service, and the article that we got for the Singapore first Omicron specific test kit, which was also lyophilized. So looking ahead, we have actually recently completed building of a new 3,000 square feet facility, which increases our lyophilization capabilities by more than 5 times. And it's not just lyophilization for PCR or MDX assays anymore. We actually open for business for quite a lot of different requests. So even food science, just yesterday I had a meeting on freeze-dried breast milk. So I didn't know that there's actually an industry for that. So people has actually approached us for various applications and requests. We are also looking to build on the strength that we have in the past 2 years for veterinary testing. So we have a BioA version of our vet test kit coming soon. A lot of it done in collaboration with LGC's reagents. And more importantly, BioAcumen Global want to take on more contract manufacturing for promising products right here in Singapore. Thank you very much.
Great. Thanks, Jimmy, for sharing some of the technical sharing on how you can make a lyophilized point of care testing. Any questions from the audience to Jimmy?
So I'm just kind of curious, moving forward for point of care, basically we are moving into microfluidics and then talking about
Yes.
nanoscales and things like that. So what do you think are the challenges in terms of doing a lyophilization in this kind of nanoscale? Do you have any experience or do you have any comments about it?
Yeah, we actually got this question before, and because each device is different, right? The only way that we can find out is usually what we'll do is we tell the customers, why don't you give us a set of your cartridges and reagents when we try to freeze dry for you. But you're correct. When you freeze dry on a microfluidics compared to say a 96 well plate, the condition and the parameters will actually be different. So we do need to go through a optimization of the primary and the secondary drying steps, right? And we may even need to go back to the starting point, the different excipients that's mixed inside, right? It does need to be optimized before we can actually achieve a good final lyophilized products. So quite a lot of work is actually needed, but I believe it's possible.
Okay, thank you. Any other question from the audience?
Eileen from Veritas. Just want to know, what is the turnaround time for lyophilization?
Oh, good question. So I get asked that quite a lot. So it varies really. From 48 hours, we have, for some assays, we actually managed to cut it down to as low as 18 hours. But typically it's about 1 to 2 days.
Depending on the volume, starting
yes,
volume.
depending on the volume. And also depending on when a customer come to us, right, whether they have done any pre-optimization work or not. So some customers, they have their own freeze dryers. They already know what's the protocol that's needed. They have already fixed their reagents. But we also have a lot of customers that came to us. So it's the original wet format. So nothing has been done to it yet. And they are using non-lyo compatible master mixers.
Probably
Commercial
glycerol and...
Yes, correct. Once you have glycerol, right, you need to start everything from scratch again. Yeah.
Thanks so much.
Thank you. Any other question from the audience? Right. If no, I'd like to again thank Jimmy for the sharing. I think that's really very important, to have that capabilities locally in Singapore to be able to accelerate the next generation of point of care testing. So with that, we have come to the end of today's session. Really thank you all the speakers once again and everyone to stay throughout the day and the sessions.